Hello, everyone, and thank you for being here. My name is Łukasz Szydłowski, and I'm working at Małopolska Center of Biotechnology, which is part of Jagiellonian University. And I would like to tell you about my project that we done in collaboration with NASA JPL, as well as Acıbadem University in Istanbul. And it will be a story about several microbial hitchhikers that got into the International Space Station, where they were discovered, sampled, and their genomes were sequenced. So we tried to find an answer of what made them survive those conditions over the prolonged time. So that is an overview of our project. So basically, samples of international biofilms of the internal components of International Space Station were sampled for microbiome, and those bacteria were then isolated. So we have single species. In this case, we got five of those microorganisms that were sequenced. And we managed to get all their protein coding sequences, which helped us to predict the structures of the proteins and functions. And we tried to look at this unique set of genes that would enable them to thrive under the conditions of microgravity, increased radiation, and all the conditions that you find on the space station. We found several sets of rearrangements in their genomes and alterations in terms of stress response, metabolic arrangements, membrane adjustments that we think that's allowed them to actually live in such an environment. So that is currently being under review in BMC microbiology. So hopefully we can get the paper published soon. Still waiting for reviewer number 2. So as I mentioned, our aims were to annotate the genomes of bacteria isolated from ISS and then compare them with their closest terrestrial relatives and identify genes involved in adaptation to the space lifestyle. So these are our isolates. So they were all new species that we found. 2 of them are coming from microbacteria, and we found also they're very closely related species that share very high average nucleotide identity, the ANI, which is over 98%. And for other species that we have, this relative ANI was slightly smaller, but they are all more or less similar, except for the one that seems to have like twice as many genes as the others. This is a spore former, Panibacillus. The rest are non-spore-forming, and all of them are gram-positive bacteria. So we did predict the gene coding sequences, and we ran several annotation tools to actually see what we can get, what information we can get from that. And we used 2 of so PGAP, which is a prokaryotic genome annotation pipeline, part of NCBI submission system. When you submit new genomes into the repository, you have to annotate them through this pipeline. Also used EGNOC, which looks at the orthologous genes, so tries to infer the gene function based on the homology to its close relatives. And we also used a tool called deepFRI functional residue identification, which is our in-house tool that our group actually developed and is working on. So as you can see, the coverage, so the information we got for using different tools varies, but deepFRI managed to pretty much annotate, give function to all of the genes that were encoded. Whereas for PGAP and EGNOG, the situation was that we had like 1/5 of the genes that were assigned as unknown or hypothetical genes, so we did not know what they truly are. So deepFRI, as I mentioned, is our in-house tool where it uses neural networks to either to predict structure and basically infer the function from by aligning it to its reference database of known structures, or we can also use just sequence itself for more generic predictions. And it's not homology dependent, so it was trained on Swiss prot models. And annotations are given either as Go terms or EC numbers, where we also receive a probability score for each annotation, so we know if this is how good our predictions are, and shall we treat it with a bit of skepticism, or we can say that these are pretty good. So this has been published 2 years ago, and it proves to be working very well on different data sets, mainly used for metagenomes, but also which are also full of species that are not very well known taxonomically. And we thought that we can apply this tool for our case study. So first of all, we looked at the common for all annotations in all of those species. So we found over 1,700 annotations, and when we applied some stringency, so we used this probability score, we then divided it into medium quality annotations, where we had at least 50% probability that the annotated function is true, or high quality annotation, where the probability was over 90 or equal to 90%. So as you can see, we reduced this number gradually as we increase the stringency as expected, and there is of course try to find some way of analyzing and sorting those annotations, trying to see what they are actually, how can they be further categorized. So we did apply this COG category. So clusters of orthologous genes have these 26 categories, which can be divided into cellular process, signaling, metabolism, or information storage and processing. So as you can see, we managed to get these these annotations positioned into assigned into those categories, and that worked really well. But this is generally, like in this case, it's a common set of annotations that you could probably find in all the other microbes, regardless if they live on space station or not. So we did next step of filtering those annotations and functional functions that those genes have. So we did for each pair of space isolates and its closely related species from Earth, we did this pairwise comparison for them, and we detected a set of unique genes in each case. So of course, more genes were found like more unique genes were found in those species that were less that had higher taxonomic distance to their closest relatives, like in case of Leifsonia. But in general, we found those genes, and then we looked at their annotations, and we found that among this unique set of genes that are not present in their closest relatives, we have about 309 annotations that are shared for all those 5 microorganisms. So we then again categorized them, and we found in this case it was a lot less, of course, as you expected, but nonetheless there were still all categories were found in this set of genes. So obviously, this is just a general overview, and we started looking at some case studies of adaptations. So we found that metallopeptidases, so geoterms related to metallopeptidases, were found in those sets of genes. So we tried to look at this what makes them so unique. And we found, for example, for one of the species, Microbacterium meriae, and its terrestrial relative, that although structurally those genes encoding those enzymes, so when we predicted the structure, they looked pretty much the same. But then when we looked at the amino acid content and similarity, they shared much lower similarity. So as you can see, this baseline is actually reference point of what amino acid content was in the protein from terrestrial species, and we see that there is an enrichment in methionines and arginines with subsequent reduction of lysines. And this actually can be referred to increasing structural stability, because arginine has three bonds, whereas lysine only makes two additional bonds. So this actually increases its rigidity. And methionines with the thiol group are also able to further lock the protein, making them less prone to to redox damage. So that is quite nice illustration of this adaptation. So in space conditions with increased redox stress and radiation, you would expect that kind of alteration of your genes to be present. Also, from the set of genes that we found that were otherwise not annotated at all, we managed to get 2 genes from those microbacteria from space station that were not present in their terrestrial relatives, where the similarity of genomes was very high, so 98 and more ANI. We found that there are 2 genes which are, according to our prediction with high quality annotation, they probably encode oxidoreductases that are located in the extracellular layer of the cell. So this again might be some interesting fact that the extracellular layer, which is primarily responsible for interacting with the environment, might be spiked with those proteins that are also might be some form of stress response or other adaptation, right? So as the genomes are not fully assembled into a single chromosome, they're still in the form of contigs. And as you can see in the contigs, we detected those biosynthetic gene clusters, both with anti-smash and our deepFRI tool, which encode pathways for secondary metabolites, also novel antibiotics, and probably more, because there is still like a large pool of those genes that are in the pool of novel genes that we are yet to fully analyze, and of course would be nice to have it experimentally validated. But so we are actually now thinking about designing some experiments to prove that those genes do have play a role in adaptation to space conditions. So in conclusions, I can definitely say that there is a convergent adaptation, even though those bacteria come from different lineages, but they get adapted to those conditions in International Space Station, and genes that facilitate adaptation to the environment may enable bioproduction of essential biomolecules needed during future space missions. So, for example, if they are also producing antibiotics, that would be nice, nice backup for any long-term missions in the future. That you have bacteria that could help you to eradicate those unwanted pathogens that could start growing inside the space station and serve a risk. And of course, because this environment is also highly mutagenic, there is also a risk that these bacteria may become a threat. And if they become a threat, at least we know what set of genes are essential for their adaptations, and we could think about designing drug targets potentially if we have to combat any any infection coming from those such kind of hitchhiking organisms. So I would like to thank our collaborators in JPL, Professor Venkat's group, Anna and Nitin, who were participating in this project, a group from Sezerman Lab in Acıbadem University, Denis and Alper, and my group from Structural Functional Genomics, Tomasz, Paweł, Paweł Szterbiak and Witold, who were also very helpful throughout this, helping to optimize the pipeline to work better. And thank you all for listening.